Welcome to this custom-tailored deep dive. We crafted this one specifically for you.
Yeah, and today we are looking at something that honestly operates almost entirely out of sight.
Right.
But it has this architectural complexity that totally challenges how we understand evolution.
It's wild. We're talking about the Coprinellus mushroom genus.
Also known as the inky caps.
Exactly. And if you've ever spent time, just walking through the woods, or maybe you've even seen these in a damp urban environment, these little delicate ribbed fungi that just pop up out of nowhere.
And then disappear almost as quickly.
Right, because over just a matter of hours or days, their gills undergo this crazy process called deliquescence.
Which is a great word.
It really is. They literally auto-digest. They use their own enzymes to just dissolve their entire cellular structure into this black, inky fluid.
And that's how they release their spores. It's a highly specialized, totally terminal reproductive strategy.
It's metal. They melt themselves to reproduce.
They do. Yeah. But the physical decay, the whole melting thing, that's really only the surface layer of what we're digging into today.
Yeah, we have a great stack for this mission. Two really fascinating sources. First, we've got a rigorous genetic analysis by James and colleagues.
From the journal Genetics.
Right. And they basically dissect the DNA of one specific species, Coprinellus disseminatus, to figure out how it essentially short-circuited its own ancient mating system.
Which is a biological mystery we'll get into.
And then our second source pivots from that microscopic lab genetics stuff to boots-on-the-ground field taxonomy.
Yeah, this is a survey by Hussein and their team, published in Mycokeys.
And they take us to the lowlands of Pakistan to uncover 4 entirely new species of Coprinellus just hiding in plain sight.
And the synthesis of these two papers, it gives us this incredible look at how biodiversity is generated. Because fungi, they so often get ignored, right?
Totally. Everyone cares about the plants and the animals.
Exactly. We see the flora and fauna, but we ignore the fungi. But Homobacidiomycetes,
which is the specific group of mushroom-forming fungi we're talking about.
Right. They possess this genetic architecture that rival and honestly sometimes surpass the complexity of animals.
Yeah.
When we look at how a species like C. disseminatus hacks a reproductive system that's been conserved for millions of years, and then we see all the physical diversity of its relatives out in the wild, we're getting a masterclass in evolution.
It's the raw mechanics of adaptation.
Exactly. It's how environmental pressures and genetic mutations all collide to solve the absolute basic problem of biology, which is survival and reproduction.
So, to really appreciate what C. disseminatus did, we have to start with the baseline of how fungi reproduce. And to do that, you kind of have to throw everything you know about male and female out the window. Right.
Mushrooms do not care about our binary framework.
No, they do not. In these fungi, reproduction is completely governed by what we call mating types.
And the ancient ancestral baseline for this is a system called Tetrapolar, or bifactorial incompatibility.
Which sounds like a mouthful, but it basically means you need 2 sets of keys to start the car.
That's a perfect analogy.
You've got compatibility controlled by 2 totally independent, unlinked genetic locations, or loci. We call them the A locus and the B locus.
Right. So for 2 distinct fungal networks to meet in the soil and successfully fuse to enter that dikaryotic phase and share cellular material, they have to have completely different versions or alleles at both of those loci.
Both the A and the B.
Both of them. And the molecular mechanics behind them are totally distinct.
Let's break that down, starting with the A locus. What is the A locus actually doing?
So the A locus encodes homeodomain transcription factors. It basically produces 2 classes of proteins, HD1 and HD2.
Okay.
When 2 fungal hyphae meet, the rule is the HD1 protein from fungus number 1 has to physically bind, or heterodimerize, with the HD2 protein of fungus number 2.
And if they have the exact same alleles,
then the proteins won't bind. Nothing happens.
It's a lock and key.
Exactly. But if they're different, they bind together, create an active transcription factor, and that kicks off the first phase of sexual development. That's the A locus safeguard.
Got it. And then the B locus is doing something totally different, right?
Completely different biochemical pathway. The B locus controls small peptide pheromones and the receptors on the outside of the cell that catch them.
These are the STE3-like receptors.
You got it. And these receptors are hooked up to this complex internal signaling system. So for the B requirement to be met, the pheromones squirted out by one fungus have to perfectly fit into the differing receptors of the potential mate.
So wait, fungus A shoots out pheromones, fungus B catches them with its receptor, and that triggers the signal inside the cell.
Yes. And it has to happen in both directions.
That is just a brilliant biological safeguard. It's so intense.
It really
And the
is.
whole point of making it this difficult, requiring a match on two totally unlinked genetic checkpoints, is to force outcrossing, right?
Precisely. The system basically mathematically restricts you from mating with your own family. Because both loci have multiple alleles floating around in the population, your chances of being compatible with a spore that came from the exact same parent mushroom as you is only 25%.
So 75% of your siblings are totally off limits.
Exactly. It actively prevents inbreeding. It pushes the organism to go out into the world and find brand new genetic material.
Which is great for genetic diversity.
It's incredible for diversity. It lets the fungi constantly adapt to new stressors, pathogens, whatever the environment throws at them.
Yeah.
The tetrapolar system is so good at this that evolutionary biologists think it probably only evolved once deep in the ancestral tree.
It's a highly conserved trait.
Right.
But, and here's where the plot twist from the James Yell paper comes, in about 25% of all mushroom species today just ditched it.
They threw it out.
They completely abandoned this complex dual locus system for what we call a bipolar system.
Meaning compatibility is now dictated by just one single locus.
And that fundamentally shifts the population genetics, right? Because if you drop the requirement for that second locus, your compatibility with your sibling spores instantly doubles.
It jumps from 25% to 50%.
That is a massive increase in the potential for inbreeding.
It is. But that transition, moving from outcrossing to a higher potential for selfing or inbreeding, we actually see that parallel across a lot of biology.
Really? Like where?
Certain angiosperms, flowering plants do it, other types of fungi do it. We're kind of conditioned by mammalian genetics to think inbreeding is always a death sentence.
Right, recessive mutations stacking up and all that.
Exactly. But in certain ecological situations, just guaranteeing you can find a mate, even if it's your sibling, outweighs the cost of low genetic diversity.
Oh, I see. If a single spore lands on a really great rotting log out in the middle of nowhere and there's no other genetically distinct mates around,
then being able to mate with a sibling spore that lands next to you ensures your lineage survives. The bipolar system sacrifices maximum genetic diversity just to guarantee reproductive assurance.
Okay, so that brings us to the subject of the first paper, Coprinellus disseminatus. This is a super common mushroom. If you've seen a massive troop of tiny little mushrooms decaying a stump, it might have been these guys.
They are everywhere, highly successful.
And they are definitively bipolar. But since we know they evolved from a tetrapolar ancestor, the whole mission of the James et al. Study was to figure out, how did they do it?
How do they break this system?
Right. And to tackle this, they looked back at a geneticist named John R. Raper, who laid out 3 hypotheses back in 1966.
The 3 ways a tetrapolar fungus could theoretically become bipolar.
Okay, let's walk through Raper's hypotheses. What was number 1?
Number 1 is mutation. Basically, one of the 2 mating loci mutates and becomes what we call self-compatible, just gets locked in the on position.
So it's constantly telling the cell, yep, we found a match, even if it hasn't.
Exactly. It totally loses its ability to discriminate mates. That's hypothesis 1.
Got it. Hypothesis 2.
Translocation. This is a physical chromosomal reshuffling. The unlinked A and B loci get physically moved on the chromosome until they fuse into one giant megalocus.
So they just glue them together.
Yeah, and we actually see this happen in reality. Smut fungi, like Ustalago hordae, do exactly this.
Okay. And hypothesis 3.
Takeover. This suggests a gradual evolutionary process where one locus just slowly assumes all the biological responsibilities of the other until the second one is totally obsolete and degrades away.
Okay, so we have mutation, translocation, and takeover. And the way James and his team went about testing these on C. disseminatus is, well, it's just amazing detective work.
The methodology is incredible.
They literally went into the Duke Forest in North Carolina, gathered these fruiting mushrooms, and they had to isolate the genetics of individual spores.
Which is not easy.
Not at all. They made the mushrooms drop spores on foil, made a liquid suspension, put it on agar, and then, using a microscope, they painstakingly cut out individual single spore colonies.
They managed to isolate 49 of these homokaryotic strains.
49. And then they crossbred them in every single possible combination.
It's an exhaustive matrix. But the data from those crosses was wild. Using a statistical model, they estimated that in just this one global population of C. disseminatus, there are around 123 distinct mating types.
123.
Which tells us this bipolar system isn't some brand new fragile thing.
Right.
It's ancient, it's well-established, and it's under intense evolutionary pressure to maintain diversity.
They also brought in some strains from Japan, right?
They did, and they found a complete reproductive barrier. The Japanese strains were totally incompatible with the European and North American ones.
Meaning they might look identical to us, but genetically they are functionally different species.
Exactly, what we call cryptic species complexes.
So once they mapped out how these mushrooms were mating, they had to actually find the DNA responsible for it, and they didn't have a fully sequenced genome to work with.
Right. This was before you could just hit a button and sequence the whole thing cheaply. They had to use positional cloning. They needed a landmark on the chromosome,
And what was the landmark?
the MIP gene. It encodes a mitochondrial protein, and in other related fungi, the active mating locus is always parked right next door to it.
It's like finding a famous monument so you know what street you're on.
Exactly. So they probed the DNA for this MIP gene, locked onto it, and then sequenced the neighborhood right around it.
And bingo, they found the A locus exactly where they expected it.
Right next to the MIP gene, a stretch of DNA with the Homeodomain transcription factors. 2 pairs of genes, AA and AB.
Now, here is where it gets interesting. To prove this was the active mating locus, they looked at the nucleotide diversity, right?
Yes. Active mating genes are under what we call balancing selection. Nature wants them to be highly polymorphic, highly variable, so everyone can tell themselves apart from strangers.
You want a lot of different lock and key combinations.
Right. And the DNA around this A locus was 10 times more variable than the surrounding junk DNA. It was definitely the active gatekeeper.
Okay, so if the A locus is doing the mating, the million-dollar question is, where did the B locus go? Where are the pheromones?
Well, if Raper's second hypothesis translocation was right,
then the B locus should be glued right next to the A locus.
exactly. But when they ran the segregation analysis, tracking how these genes were inherited by the offspring, the B locus markers were completely unlinked from the A locus.
They were nowhere near each other.
Nowhere near each other. So Raper's translocation hypothesis is completely busted for this species.
So if it didn't fuse, where is it? Because it wasn't next to the landmark, they had to go fishing for it.
They used a technique called degenerate PCR. It's basically genetic dragnet.
How does that work?
Since they didn't know the exact sequence of the missing B locus, they designed primers based on the known, highly conserved B locus structures from other related tetrapolar mushrooms.
So they threw out a net hoping to catch anything that vaguely looked like a pheromone receptor.
And they caught it. At least 4 distinct pheromone receptor genes and 3 small peptide pheromone genes. They found the physical architecture of the missing B locus.
It was just floating somewhere else in the genome, but what state was it in?
This is the key. Remember how the A locus was hypervariable, 10 times more diverse?
Because nature wants lots of mating types.
Right. Well, when they sequenced these newly found B genes across all these different isolates, the diversity was basically flat.
It had dropped down to just the baseline mutation rate of normal, everyday cellular housekeeping genes.
Exactly. There was 0 balancing selection. These genes were completely free from the evolutionary pressure to act as a mate discriminator.
They were basically demoted.
Entirely demoted. They still had their structural coding, but they were not acting as a checkpoint for reproduction anymore.
Okay, so they found these demoted zombie genes. The next logical step, which is frankly my favorite part of the whole paper.
The Frankenstein experiment.
Yes, heterologous expression. They took these demoted B genes out of the Bipolar disseminatus and shoved them into a completely different mushroom.
Specifically, Coprinellus disseminatus, which is a strict, old-school tetrapolar species. It still requires both A and B to mate.
So they put the demoted genes into a host that actually uses those genes. What happened?
The genes woke up and worked perfectly.
That is insane.
It is.
Yeah.
If Raper's third hypothesis, the takeover one, was correct, you'd expect these genes to just be broken, useless junk. But they weren't. They immediately interacted with the host's internal machinery.
It started forming clamp connections.
Right, those little backward-facing branches that make sure every cell gets both nuclei. It formed subapical pegs.
And the craziest part, when they crossed it with a compatible tester strain, it grew into a full, mature fruiting body and released viable spores.
The whole B locus machinery from C. disseminatus was completely intact and functional, just wasn't being used to initiate mating in its original body.
So that completely rules out the takeover hypothesis. The A locus didn't slowly replace the B locus.
Right. So by process of elimination, we arrive at Raper's first hypothesis,
Mutation.
mutation. Somewhere deep in the evolutionary past, a mutation happened in the bee locus that made itself compatible. It got stuck in the on position.
And the researchers actually think they found the exact mutation, didn't they?
They did. They found a specific amino acid substitution in one of the receptors, CDSTE 3.1. In the third cytoplasmic loop of the receptor, a phenylalanine residue had replaced an aliphatic amino acid.
And why is that significant?
Because we see that exact same mutation in baker's yeast. And in yeast, that specific swap acts like a jammed gas pedal. It locks the receptor into a state of continuous activation.
So it's constantly screaming at the inside of the cell, we have a pheromone match, let's go!
Yeah.
Even when there are no pheromones around at all.
Exactly. It completely bypasses the B checkpoint. And because the B requirement is permanently satisfied in every single spore, all the pressure to discriminate mates falls entirely onto the A locus.
That is how a two-lock system becomes a one-lock system.
Tetrapolar becomes bipolar.
But wait, this brings up a really big biological question. Maintaining complex cellular receptors, making these proteins, managing the internal signaling cascades that costs a lot of cellular energy.
It's expensive.
Very expensive. If the B locus is permanently jammed in the on position and it's useless for finding a mate, why hasn't evolution just deleted it? Why does C. disseminatus keep these functional pheromone genes around for millions of years?
That is the profound realization of this paper. We have to reconsider what these genes are actually doing. In these Homobasidiomycetes, we've never actually detected these pheromones operating outside of the fungus.
Wait, really? They don't broadcast them into the soil?
No, it appears to be entirely internal. The current theory is that while these genes lost their job as the initiator of mating, they have an absolutely indispensable secondary job, maintaining the decaryon.
Remind me, the dikaryon is that phase where the two nuclei are just living together in the same cell without actually merging?
Right, they cohabitate. And as the fungal network grows, those two distinct nuclei have to divide in perfect synchrony. Managing that dual nucleus state requires immense biochemical coordination.
You have to build cross walls, form those clamp connections, keep the nuclei spaced properly.
Exactly. And the internal pheromone system is basically the communication network running that structural maintenance.
So the system wasn't preserved to find a mate, it was preserved to manage the household after the marriage happened.
That's a great way to put it. It shows that evolution rarely just deletes complex systems. It repurposes them. The function just shifts based on what the organism needs to survive.
That idea of biological architecture adapting to shifting demands, that is the perfect pivot to our second source.
The Hussein et al. Study.
Yeah. While James and his team were zoomed in on the invisible DNA, the Hussein study is looking at how all this genetic variation actually physically manifests out in the real world.
We're moving from the lab to the field, specifically to the subtropical and temperate lowlands of Pakistan, the Khyber Pakhtunkhwa and Punjab regions.
And the context here is really important. Fungal biodiversity in Pakistan, especially for these these inkcap clades, has been hugely underdocumented.
Completely. Before this 2018 study, there were only 18 coprinoid species officially known in the entire country.
Which is wild because the climate in those lowlands is basically a paradise for saprotrophic fungi.
Right. Saprotrophs are the decomposers. They eat dead organic matter. And these forests have an endless supply of decaying wood, leaf litter, animal dung.
So mycologists knew there had to be more species out there just waiting to be categorized.
And Hussein's team proved it. They found 4 entirely new species of Coprinellus. And the descriptive taxonomy in this paper is just incredibly detailed.
You really have to zoom in because to the naked eye, a lot of these little brown mushrooms look exactly the same.
Exactly. So let's look at the first one, Coprinellus campanulatus.
They found this one growing in clusters on woody litter under white mulberry trees, right?
More cell, but yeah. And visually, it has a campanulate or bell-shaped cap. It's grayish olive with a dark yellowish brown center. But its standout feature is this micaceous veil.
It's literally glistening. It looks like it's covered in bright white mica.
Right. And microscopically, that glistening effect comes from these loosely arranged, perfectly spherical globose cells sitting on the cap.
But to really confirm it was a new species, they had to look at the spores.
Spore morphology is classic taxonomy. The spores of C. campanulatus, when you look at them face on, are mitriform.
Meaning they're shaped like a bishop's mitre, like a little pointed hat.
Exactly. You measure the dimensions, the geometry where the germ pore is. That's how you separate species that otherwise look identical.
Which brings us to the second discovery, which has a very familiar name. Coprinellus disseminatus similis.
The similis tells you right away it looks incredibly similar to the C. disseminatus we were just talking about from the genetics paper.
They found this one in leaf litter under poplar and mulberry trees. It starts out as this little white cylinder, then expands and turns grayish-brown right before it melts.
But the rigorous microscopy is where the differences emerge. Macroscopically, it has an umbononate center.
A little bump right in the middle of the cap.
Right, which standard disseminatus doesn't usually have. But more importantly, the microscopic hairs are totally different. It has these massive, uniquely shaped sterol cells on the gills called chylcystidia.
And the cap is covered in narrowly utriform pileocystidia, which are basically microscopic hairs.
Right, and the original C. disseminatus does not have those specific pileocystidia. So structurally, it's a distinct species.
Yeah. Species number 3 is Coprinellus pakistanicus. And this one lives in a totally different micro-niche.
Yeah, it wasn't on wood or dense leaf litter. It was scattered on moist, bare soil under acacia trees.
Acacia nilotica and Acacia modesta. Visually, it's pretty different too. Light yellowish green to grayish yellow and totally smooth. No glistening veil, no microscopic hairs.
And it has a depressed center, not a budding one. But what makes Pakistanicus really cool is its DNA. When they sequenced it, it didn't group with the usual suspects.
It carved out its own entirely independent evolutionary branch.
They called it clade III right next to the subilical group.
Yeah.
And microscopically, its spores are broadly ellipsoid, not pointed like the others. It's a highly divergent lineage.
Which leaves the final discovery, Coprinellus tenuis, found on the leaf litter under Acacia modesta.
Tenuis means thin or membranous in Latin.
Which makes sense. It has this incredibly fragile, deeply pleated cap, like radial grooves running almost all the way to the center.
And it's glabrous, meaning bald, but structurally it looks kind of furred. But the main diagnostic feature of tenuis is the sheer size of its spores. They are massive compared to its relatives.
And its closest genetic cousin is a species called Coprinellus curtis, right?
Yes. And this is a perfect example of sympatric speciation. They share a lot of physical traits, like that deeply pleated cap, but their ecological jobs are completely different.
C. curtis is a dung lover. It only grows on herbivore poop.
Coprophilous, exactly. Whereas C. tenuis evolved to break down the tough lignocellulose in terrestrial leaf litter.
So they live in the exact same geographic area, but they don't compete for food. They carved out different metabolic niches.
Exactly. Now, documenting all these microscopic hairs, the veil shapes, the spore geometries, this is how mycology has operated for centuries. It's called the Frisian system.
Right, organizing everything by what it visually looks like.
Right. And under that traditional visual system, the Coprinellus genus was divided into 3 main sections based on the cap.
You had the Domestici section, which had these floccose, scaly veils.
And the Mycaceae section, which had that granulose, glistening, mica-like veil.
Like the first one we talked about, C. campanulatus.
Right. And then you had the Setulosii section. These were the ones completely covered in those microscopic sterile hairs, the pileocystidia.
It makes sense, right? Group them by the obvious physical traits. But, and this is the major plot twist of the Hussein paper, modern DNA sequencing comes in and totally destroys that visual classification system.
Completely rewrites the rulebook.
They extracted the DNA from these new species and sequenced the ITS region.
The internal transcribe spacer. It's the universal genetic barcode for fungi.
And when they built a new evolutionary family tree based purely on the ITS DNA, the physical traits didn't line up at all.
Not even a little bit. The new species C. disseminatus similis is the best example of this.
Right, remember we said it's covered in those microscopic hairs, the pileocystidia?
Under the traditional visual system, that means it unequivocally belongs in the Setulosii section.
But genetically?
Genetically, the ITS data drops it directly into the middle of the Mycaceae clade.
Next to a bunch of mushrooms that don't have those hairs at all.
Exactly. It has the body of one group, but the actual genetic blueprint of another.
So how does that happen?
Convergent evolution. It proves that these complex physical traits, like developing tiny hairs on your cap to deal with moisture, or maybe to stop microarthropods from eating you, that didn't just evolve once in a single common ancestor.
The environment made that physical trait so useful that totally different genetic lineages evolved the exact same solution independently.
Nature arrived at the same architectural blueprint using different paths.
That is huge for taxonomy.
Massive. It means we cannot trust our eyes anymore. If convergent evolution can make unrelated mushrooms look identical under a microscope, then organizing them by how they look is always going to give us a flawed evolutionary history.
It's a total paradigm shift. We have to move from what it looks like to what the DNA actually says.
The physical traits are still important if you're out in the woods trying to identify something, but they have to be contextualized by the genetic reality. The old visual categories were basically just artificial constructs.
It's wild, and it really brings this whole deep dive full circle. We started down at the absolute bottom, looking at the invisible DNA architecture with James et al.
Watching Coprinellus disseminatus literally short-circuit a millions of years old mating system.
Just one tiny amino acid substitution, a phenylalanine swap, and suddenly a tetrapolar species becomes bipolar. It embraces inbreeding.
And yet, it meticulously keeps the old pheromone receptors around, not to find mates, but just to manage the internal plumbing of the cell.
Right, maintaining the decaying. And then we zoomed all the way out with the Hussein paper to see how that massive underlying genetic creativity actually bursts out into the physical world.
Seeing saprotrophic fungi adapting to every possible micro-niche in Pakistan, evolving new spore shapes, new veils, new cellular hairs, and completely defying our attempts to easily categorize them.
The Coprinellus genus really is a perfect microcosm for biology itself. Evolution is not this straight, rigid line. It's messy. It's constant problem solving and repurposing.
Exactly.
It definitely changes how you look at a cluster of inky caps just melting on a log. You're watching millions of years of overlapping evolutionary hacks playing out in real time.
It's profound. And honestly, it leaves us with this one really compelling, almost unsettling thought about genetics.
That's that.
Well, we just saw absolute proof today that an organism can maintain a highly complex, metabolically expensive pheromone communication system, keeping it completely intact while totally stripping it of its original reproductive purpose.
Yeah.
The genome is basically a historical archive. So if a simple autodigesting fungus is capable of that level of profound genetic repurposing, you have to ask yourself, what other ancient, entirely unseen genetic ghosts are silently driving the biology, the cellular maintenance, and the instinctual behaviors of the plants, the animals, and even the humans interacting in the ecosystems around us right now?
Man, that idea of ancient biological echoes just silently pulling the strings today, that is definitely something to sit with. Thank you for joining us on this deep dive into the hidden complexities of the fungal world. Keep questioning the unseen architecture of the natural systems around you. We'll see you next time.